It's my pleasure to introduce our speaker today. Dr. David Ellis has been involved in the conservation of plant genetic resources for decades, with experience in academia, private industry, and the public sector. He was the head of the USDA Gene Bank at the National Center for Genetic Resources Preservation in the United States prior to retiring as the head of the Gene Bank at the International Potato Center in Lima, Peru. His research interests have spanned plant development, medicinal compounds in plants, plant molecular biology, plant and insect ecology, cryobiology, and conservation of plant diversity. He is currently active in the collection and conservation of sweet potato landraces from Africa and in launching the Global Plant Cryopreservation Initiative. The title of his talk today is, When is it good to be left out in the cold? The Global Plant Cryopreservation Initiative. Welcome, Dave.
Thank you, Yanni, and thank you, Faye, and thank you everyone at the Crop Trust and CGIR for this opportunity to meet with you all this morning, and thank you all participants for joining us. What I'd like to talk about today is an exciting new initiative which we hope will complement the Svalbard Global Seed Vault in Norway. And the attention today will be on those genetic resources collections which cannot be maintained as botanical seed. And I'd like to set the context of my talk today by starting with a little make-believe story, if you'll entertain me with that. So let me take you back to the day, but a day when none of our immediate ancestors were alive. Imagine us back 15,000 years ago. And what we are, our ancestors are these hunter and gatherers. And it's a tribe that's been in a very lush valley for the last days or weeks, and it was time to move on. And as they moved on, one member of the tribe who was invalid in some way, maybe a broken leg, maybe a bad toe. And again, now it won't advance for me. There we go. Couldn't keep up with the tribe, and the tribe just had to move on. They didn't notice this one individual missing from their group because their survival, they worked on their own. So this individual was left in the valley alone when the rest of the tribe moved on. Fortunately, he or she was able to survive, picking low fruit, picking easy to find seed that maybe was a little drier than previously desired by the tribe because they had fresh seed and nice moist seed to eat. So he or she made a subsistence, this ancestor of ours, and outside the cave, as he ate food and distributed it, a refuge pile started to build. Now, alas, our ancestor didn't make it through the winter there, but over the next several hundred years, this scenario was repeated time and time and time again. And from this refuge pile, new plants started to arise. And these plants were a little different from those that were in the valley generally. Maybe the fruit was a little bigger. Maybe the plants had fruit that was lower or easier to pick. This was the start of the use of diversity by humans to advance and to start domestication. Now, this concept, the use of diversity, is not new, and most of you are students of Darwin and have read on the origin of species and know it as a treatise on natural selection. But how many of you know that the alternative title of the Origin of Species, and here you see the cover page of the original document, and it says right there, the alternative title is Preservation of Favored Races in the Struggle for Life. And in favored races, Darwin recognized not only natural selection, but also artificial selection. And he saw the power of both of them, Conjuring up Darwin always puts a tickle in my throat. As a matter of fact, in the very first chapter of Origin of Species, Darwin goes on to say that although the process of selection by man can do much, and he sees no limit to the amount of change to the beauty and infinite complexity, no amount of change to that diversity that can happen with one another in their physical conditions of life. Thus, he recognized the power of both natural selection, which we all know from Origin of Species, but also the power of artificial selection. And he went on to say the appearance of new forms and disappearance of old forms, both natural and artificial, are bound together. Now, this is where we're having a slight departure from Darwin, because we know, as Darwin did, that diversity is critical. However, current rates of disappearing of plants is going on at an alarming rate. A recent estimate was that 2.3 plant species disappear annually. 2.3. That blew me away. That's a lot. And that the current extinction rate of plants is 500 times greater than the natural or background extinction rates for the last 1,000 years. That means although it's natural, as Darwin said, for plants to disappear and appear, we are going through a period where the extinction rate of plants is exponentially higher than what we've ever experienced before. And why is this important? Is because this diversity has been used by humans to do great things. I just use a few examples here. The first one is Norm Borlaug with the Green Revolution and the rice variety of Norin 10, which was derived from germplasm, bred from germplasm from 3 different countries. If we didn't have the diversity there from the 3 countries, we may not have had a green revolution. We may not have had the impact on feeding a quarter of the world's population. A good example from a wild rice species is that this one wild rice species has been the source of grassy stunt virus resistance in tropical rice for 36 years. And it continues today, slowly breaking down, I hear. But we still, if we didn't have this rice species, and if we didn't have the diversity that had this resistance gene, rice may have been succumbing and not be as productive as it is today. C88 is a very famous late blight resistant potato. Its pedigree has at least 17 different accessions. If any one of those accessions were there, perhaps C88 would not have been bred and would not have given the benefit to poor farmers around the world of almost $200 million a year. A final one is a wheat variety called Veery, which has been used extensively in breeding programs for drought resistance. It was derived from 3,000 crosses from 51 accessions from 26 countries. Now, if this diversity had disappeared, perhaps none of these great accomplishments in breeding would have been possible. Now, how do we store diversity? We know that plant diversity is mostly stored as botanical seed in gene banks. And what I'm showing here is a picture of just a few of the gene banks that I have visited. And they come in all kinds of shapes and forms, from very rudimentary in freezers, plastic containers in freezers, all the way up to extremely advanced. This is Fort Collins, and another picture of cryotanks. Seed can be stored very well in these environments. And the gene banks are incredible places to visit. They're a wealth of diversity. I think everyone should take their kids sometime to go visit a gene bank, because this is what we need to have future generations recognize the importance of storing diversity and the importance that gene banks make. There are some poignant stories of the gene banks, but they point to the dedication of people that work in the gene banks. And I think the best example is from the Soviet Union Bureau of Applied Botany, or better known to all of us as the Vavilov Institute. Do you know that Vavilov, over 20 years, did 115 expeditions to 64 countries and collected over 380,000 samples? Now remember, this is in the 20s to the 40s, when traveling around wasn't so easy. A modern day explorer. During the siege of Leningrad, which lasted 872 days, they estimate that 1,500,000 people died, many of them due to starvation by not having enough food to eat. The seed that Vavilov had collected that was stored in the Vavilov Institute, or the Bureau of Applied Botany, as it was called in those days, could have helped some of those people sustain life, probably just for days or weeks, but probably not completely. However, there are stories of people working in that gene bank that did not eat the seed that they were saving. Probably the best known is the curator of rice, died at her table with rice seeds sitting in front of her, died of starvation. People working in gene banks are dedicated and extremely productive people for what they're doing. Further, it's impossible to assess the future economic value of one accession versus another. This is a picture of sweet potato. And how do we know that this sweet potato here up top will be any more valuable than this one later in life? We don't. That is why it's important to store all diversity and protect it well. But gene banks themselves are also vulnerable. We know the story of the gene bank in Afghanistan. This is a picture of it. When the Mujahideen came into Afghanistan, they deemed the gene bank unnecessary and closed it down. And when the Taliban took over, they completely destroyed it and set it to fire. And this is what's reportedly left of the gene bank after the Taliban got done. But it's not only man-made disasters, it's also natural disasters. There was a typhoon in the Philippines that wiped out the Philippines gene bank. I remember seeing an email or letter from the curator of the Philippine gene bank that talked about growth chambers and refrigerators floating around in the water. Gene banks are vulnerable. We hear regularly about losses of field and greenhouse genetic resources collections, but they're not safe even in a gene bank. And thus, the FAO has deemed that, has set a standard for gene banking where all collections have to be backed up at least one location distant from the primary gene bank. This is the genesis of the Svalbard Global Seed Vault up in Norway. This, in my view, the Svalbard Global Gene Bank is perhaps one of the greatest things that have occurred in my lifetime. It's certainly up there in one of the top 10 things that we have done to preserve diversity. The Svalbard Global Gene Bank currently has over 1 million samples from almost 100 different countries. It's an extreme success, and it was born due to the vision of some very brilliant people that saw the need for this kind of thing. However, let me put it in context, is that there are 50% of the 10 most important crops to humans that cannot be stored in Svalbard because their genetic resources collections cannot be stored as botanical seed. Thus, Svalbard is tremendously successful, but is not storing all of the diversity that we need today to be conserved, to keep the legacy alive for our ancestors, that they have the same opportunities to use this diversity as we do today. Because of this, because of these crops, important crops that cannot be stored in Svalbard, a study was done in 2017 by the German, Australian, and Swiss governments, funded by them, to investigate the feasibility and need for a safety backup facility for those crops that could not be stored in Svalbard, cannot be stored as botanical seed. And these include clonal crops, because we want to keep the clonal crops, the genetic integrity, the makeup in the clonal crops intact. That's what's important for the clonal crops. They do not breed true to seed. We've got other seed, plants that produce seed that cannot be dried down. In order to store in Svalbard at -20°C, the seed has to be dried down, or the water content causes the seed to die. So you need to dry seed down. If the seed can't be dried down, this is termed recalcitrant seed, the seed does not survive when stored. And there's other seed that's short-lived seed that maybe will only store 10, 15 years at -20°C. These are candidate seed for a alternative storage form. We have no Svalbard equivalent for these, and the genetic resources collections are at risk. And so this study, you have a link to this study. If you haven't seen it, please go and look at it. If interested, you have a link to this study in your background information for this webinar. Now, the conclusions from the study were, number one, that cryopreservation is the best long-term conservation option for clonal and recalcitrant seed crop collections. Makes sense? We have no other option currently, and so this is the best for long-term storage. Cryopreservation has lower running costs, increased longevity, and greater genetic stability of collections stored this way. But the study also recognized that there are high initial costs and skill and technical challenges to running a cryopreservation gene bank. And we'll talk about those further during this talk. The study also concluded that cryopreservation has huge benefits. It's not only the best long-term conservation method, but it has huge benefits for long-term backup. When it's compared to in vitro, which is what the next best storage method they deemed was, in vitro is much more costly, the material is difficult to transport, and it needs to be continually replaced. And with cryopreservation, although you need infrastructure, the needs for a backup gene bank were relatively modest, because they estimated that only about 5,000 accessions currently need to be backed up, currently are in cryopreservation, but that number should double in 10 years. I would say we've already surpassed the 5,000 accessions now in the last 5-6 years, and that perhaps we would look at tripling to 15,000, maybe 20,000 accessions in the next 10 years. The need is there. Where the need really is to accelerate the development and implementation of operational cryopreservation methods. And we'll talk about what I mean by operational cryopreservation. And we need to do this to safeguard these clonal and recalcitrant crop collections. The feasibility study estimated that there are currently 100,000, if not more, Annex 1 accessions, Annex 1 from under the International Treaty for Plant Genetic Resources for Food and Agriculture, that are currently at risk in field or in vitro gene banks. These are the prime candidates that we need to back up before they are lost forever. And the study also concluded that the CGIAR was ideally positioned for proposal development and seek donor sponsorship. And why the CGIAR in particular? For many reasons. One, they have existing cryopreservation infrastructure. A few of the gene banks at least do. And they are positioned in the developing world where the need is greatest to protect these in the field and in vitro gene banks. So plant cryopreservation today, we have no uniform standards or guidelines. Every cryobank operates on their own. Yes, we do have meetings where we talk about how we do things, but by and large, we have no guiding principles. And I'm not a fan of standards, as everyone knows, so I would say we'd want some guidelines rather than standards. But we do need something, and we've got to start the conversation. Gene bank managers are adverse, like all of us, to throwing anything away. You have dead seed. Oh, it may be of use in the future. How do I know this diversity is securely conserved elsewhere if we're calling it a duplicate? And knowledge will improve and future generations will be able to regenerate. These are quotes. Some of them I have used, some of them I hear regularly by gene bank managers. And my question to everyone is, what will our great-grandchildren find in our cryobanks? We asked that question at the International Potato Center in 2013, 2014. We pulled out vials from every accession in the cryobank, from potato and sweet potato, and we threw away 50% of the accessions in the potato cryobank and 90% of the accessions in the sweet potato cryobank. These collections, fortunately, we had all of the accessions in vitro, but what we had in cryo was non-regenerable, was contaminated, or dead. It was a very difficult operation to make, but we had to start at a point where we could move forward with a cryobank we were proud to leave to our grandchildren. And that's exactly what the International Potato Center has today. I know and I fear from almost 30 years working in cryo that most cryobanks are storing subpar material. You may ask why? Why bother? Because pre-1980s, we kept anything that survived. Anything. This is where the famous quote came from that, oh, future generations will be able to regenerate it. I am not so sure that even now we can regenerate much of that material that contains simply cells that we put into cryo. Through the 80s, we kept anything that we looked like a leaf. It was green. This material as well probably is not regenerable even today. Through the 90s, we advanced to wanting to keep anything that looks meristematic. Now, I like that. I think this material might have a future. However, we needed to do a step further, and today, the guidelines that I hope everyone doing cryobanking is using, that we don't put anything in the cryobank that we cannot regenerate a normal-looking plant with roots that will survive ex vitro. And this is the standard that we went to at the International Potato Center and is being used at most of the global gene banks today. The problem is the cryobanks are full of this stuff. And full is not the right word. However, if you look at the example I gave of sweet potato, it was full of material that wasn't regenerating and subpar. So let's ask the question, will future generations know what's in our cryobank? We have vials in there stored with various purpose and varied numbers. Quality standards continue to evolve today. What I hope to address through this initiative is that all cryobanks in the world embrace superior quality standards. Let's admit what we have. Yes, it takes resources. Yes, it takes time to go through vial by vial out of your cryobanks, but it's needed. Garbage in, garbage out. That's exactly what we have in the cryobanks today. We've got to admit it and move on so that we're leaving a legacy. And so I ask, when was the last time you did housekeeping and cleaning of your cryobank? I think maybe it's time to start planning that. If not now, at least within the next 5 years. I look at cryobanks today, plant cryobanks today, having 3 categories of material. The first category is fragments of research or archive material. These are leftovers from protocol development and research. I do not consider this cryobanking of plant genetic resources collection. I consider this keeping old research material that some scientists long retired ago threw in cryo and no one has cleaned it out yet. It's time to start culling that stuff out so we're not leaving it for the next curator to ask, what do I do with this? The second category are remnants from initial cryobanking. This category of material is often tied to very short-term funding. So you may have 2 years of funding, so you just do a little bit. Again, it's research material. The objective is cryobanking, but it falls short. I guess my main concern with these materials is that they have a low number of accessions, a limited number of vials, which makes this cryobank not an operational cryobank, meaning with a limited number of vials, you pull out 2 vials, you have 1 vial left. Is that really a good cryobank? You don't even have enough material for a safety backup. Material in the cryobank, the exact same accession, could have been cryopreserved by different methods. So the cryobank lacks uniformity and predictability. How is a future gene bank manager of this cryobank going to know what vial was put in by what method? We've got to clean it up. Data in the database is minimal, if it's present at all. And again, the objective of these cryopreservation, this cryobank, was to show the ability to do cryo, to attract more funding, and to get a publication. I would argue that neither of these categories is good for the long-term conservation of plant genetic resources collections. The third category is what I'm calling operational cryobanking. This is what we have to strive for in our plant genetic resources collections. Cryobanking of accessions of a particular crop that we're putting the entire collection into cryo. Collections that are hundreds, thousands of accessions, and we're getting them all in. Putting in 10 accessions out of a collection that's 1,000 accessions is not really a good use of your cryobank, and it's not an operational cryobanking. It's putting a few accessions in, and how do you know that you're putting in the right accessions for future use? The focus on cryobanking has to be future use and preservation of the diversity. We should have written guidelines with written operational protocols that include how viability assessments were made so that future gene bank managers can use the same regeneration protocols that we did to put this in place to ensure that we're not losing viability of the material. We need uniformity because uniformity will give predictivity to future generations. If we say you get regeneration of 30% of the shoot tips that you put into cryo or 30% of the whatever you put in, somatic embryos, we need to make sure that a future manager of that gene bank can get that same viability that we did. And the only way to do that is to have written protocols that they can look at 20, 30, 50, 100 years from now. Remember, these things last for centuries. Every vial needs to be documented in a permanent database, listing the methods, listing the viability, listing the person responsible. And labels in the cryobanks should be printed, nothing handwritten. So what I call are points to consider in plant cryo when we're looking at doing it from a feasibility standpoint. Cryo has been reported with over 40 different crops, and this is probably a way underestimated, maybe 100 different crops. Why then, if you've got so many crops that have been in the literature that have been cryopreserved, do we only have a handful of crops with 100 or more accessions in cryo? The reason is that the focus has been short-term funding to publish a few genotypes, publish on a few genotypes successfully in cryo, and not the entire collection. We need to change the dogma to start looking at entire collections going into cryo. There is a critical need as well to secure plant genetic resources collections long-term that cannot be stored as seed, and cryopreservation is the way to do that. And this is where I talk about it. A large range of tissues can be cryopreserved. Pollen, seeds, short shoot tips, dormant buds, cell suspensions, embryogenic material, somatic and zygotic embryos, callus. One size doesn't fit all. I think we need to recognize in setting up guidelines for cryopreservation that they need to be flexible. They need to evolve so that we can work with them in an operational setting. Unfortunately, today, most cryo methods, exception of pollen, seed, and dormant buds, require in vitro. And I call this an opportunity and a curse because in vitro can be the limiting factor. Not always, but can be. Material should be genotyped, and disease-free starting material is critical. And not everyone needs to actively do cryo. We can work with long-term commitments of funding and work with other groups to do cryo. I've heard cryopreservation is too expensive, but unfortunately, I don't think that's true, or fortunately. Yes, you do need infrastructure. Yes, you do need secure liquid nitrogen, but it is a heck of a lot cheaper than in vitro in the long term. There is a large upfront cost, but it's not a long-running cost. And the other question is, how do we know cryopreserved material can live for centuries? Nothing lives forever. Plant cryopreservation has been around since the 1970s, and it's easy to mess up. But the theoretical half-life of cryopreserved material is a long time. And there's a reference by Walters that's hidden here by my screen that lists the half-life of lettuce seed for over 500 years. I think we can depend on cryo, if done right, to last for centuries. And remember, what we want to do is we want to leave almost absolute predictivity for the future. You look at this gentleman here on the side, does that look like all of us today? We all have far too much going on. And that's not going to change, so let's make it easy for them in the future. And to put into cryo, we want identity verified and phytosanitary clean material so that it can be easily used by whoever pulls it out. Cryo programs are increasingly important. We increasingly, I've gone over this need long-term protection of diversity. I mentioned that 5 of the 10 most important crops for humans are vegetatively propagated and collections are not able to be stored in Svalbard. Potato, cassava, sweet potato, yam, and banana are those 5. And that all plant genetic resources collections that cannot be stored long-term as orthodox seed are at risk. This includes food crops, horticultural species, medicinal plants, and trees. So what is the vision that we're trying to look at for these crops that cannot be stored as seed? let me introduce to you the Global Plant Cryopreservation Initiative. What this initiative would do is focus on recalcitrant and clonal crop genetic resources collections in the developing world. Why the developing world? Because that's where the need is greatest, and that's where the funding need is greatest. But we do need support from all. It would be based, the initiative, and I'll go over this shortly, based on regional centers of excellence to provide capacity building, know-how, awareness, and support for putting these collections into cryopreservation, and provide a network for global plant cryo and safety backup of cryo collections. So what we'd like to do is get a Svalbard-like group of hubs that can store the backup of cryo collections for millennium and for the world. The vision has to be for centuries. This is what's unique about cryo. We need to ensure that we leave usable genetic resources collections for our grandchildren. We need highest quality. We need to do viability monitoring. Today, cryopreservation and cryogene banks don't routinely have monitoring, yet it's a requirement for seed collections. We need operational cryopreservation protocols, and we need a safety backup. And for GANI, you will know that we need a quality management system for gene banks. Again, we need guidelines that provide flexibility, but we need to have key points of quality management. And I think with minimum guidelines, cryo will provide what we need for clonal and recalcitrant seed collections. The current model for the Global Plant Cryopreservation Initiative is based on cryopreservation hubs. These would be specialized hubs that contain existing infrastructure, expertise, ongoing cryopreservation programs. They're locally, regionally localized, and have access to partners in the developing world. For this reason, the three hubs that we're basing our model for the cryopreservation initiative are Alliance Bioversity in Belgium, which has the Intrust Banana Collection, an Americas Hub, which is the International Potato Center, which has the Intrust Potato and Sweet Potato Collections, and an Africa Hub, which would be at IITA in Nigeria, which has an Intrust Cassava, Yam, and Banana Collections. All three hubs currently have cryopreservation ongoing and operational cryopreservation ongoing of collections. The hubs will provide expertise for the development of methodologies, move research results to operations, capacity building, services for operational cryopreservation for those that cannot do it, and cryosafety backup. As well, they will coordinate and maintain the development of a network, a cryopreservation network. The structure will not be static because we're trying to look into the future now. It will be dynamic and will evolve. Hubs may be added, modified, or changed based on need, crops, or donor preference. We currently have 10 initial crops targeted for the initiative. 7 are Annex 1 crops, again, Annex 1 under the International Treaty for Plant Genetic Resources for Food and Agriculture, and 3 non-Annex 1 crops. 7 of the crops are clonal, and you can see the crops here. 3 are recalcitrant seed crops. Our vision is that materials will be available. Anything in this initiative will be available through the Standard Material Transfer Agreement of the International Treaty for Plant Genetic Resources, or for nations that are not party to the treaty, under a similar material transfer agreement as the international treaty, but under Nagoya as a bilateral agreement. And the focus will be on collections, not research. I mentioned that this hubs will be doing some research to optimize protocols. That's going to be necessary. However, we're going to focus on getting collections into cryo and building on the success that we have today with potato and banana. These specialized hubs will be the foundation for a network, a plant community that will create a plant community of practice. They will be comprehensive networks to include national programs as well as any interested parties. And we already have a couple of networks up and running. The European Union has the Plant Genetic Resources Cryo Working Group. And just yesterday morning, we had a meeting with the Latin America Plant Cryo Network that included 12 different countries in Latin America. Very productive meeting. These networks are currently started and ongoing. The initiative will provide a database of clonal and recalcitrant seed collections so everyone can see what is there and where the need is. We'll have directed workshops, the first one happening last year in Peru, again for Latin America, to conduct and monitor the status of the collections and the status of the need for the collections to go into cryo. The network will provide a forum for sharing experiences and provide capacity building and coordinate movement of the phytosanitary clean materials. And I say, if not now, when for a cryo initiative? Where do we stand currently with it? The proposal and summary have been drafted. We're currently active in a campaign for raising funding. We have an initial project funded by the Darwin Initiative out of the UK for the collection of sweet potato land races from Madagascar and from Zambia in partnership with the Seeds of Resilience Project, where this material is currently being transferred to Lima, where it will be put in cryopreservation. The Plant Cryopreservation Network for Latin America is being developed, as I mentioned, and SIP is currently building a new state-of-the-art cryobank in preparation for this cryopreservation initiative so that the capacity is there. And capacity building programs are ongoing in all three of the hubs, Bioversity in Belgium, IITA in Nigeria, and SIP in Peru. So with that, let me thank you for everything you all are doing for the future generations in supporting cryopreservation for the backup of genetic resources collections. And you will see a pop-up on your screen. It will only be up for about a minute after the presentation for you to indicate if you're interested in staying informed on the Global Plant Cryopreservation Initiative as it is launched. And with that, I think I will turn it over to Yanni, and hopefully we have a few questions. If not, perhaps we can dance around and sing. I don't know. Yanni.
Thank you very much, Dave. That was excellent. I love that you walked us through the different challenges and the evolution of it, and why what we have today needs to be inspected, as well as developing things in the future that set a standard. So in front of you, you can see the poll. We just want you to indicate a yes or no, whether you would like to stay informed about the Global Cryopreservation Initiative. Please say yes or no, and then we can continue. We do have one question, and actually it's a two-part question. So once you answer that, I will go ahead and greetings to Vanya, who has submitted one question for you, Dave. And it is the following. When you have things in vitro, it might require more than one season for the accession to recover once it's brought from cryo, into the field. She's talking about the efficiency of bringing plants back from either in vitro to the field or cryo to the field. So it will take a while if you bring things from in vitro to the field. And her question is, what about cryo? Would it take longer to complete the wake up of these plants once they're in cryo and you're bringing them out to the field? That is the first question. And could the cryopreservation for too long of those accessions make the accession not recoverable in the field? So it's kind of a two-part question.
Yep, Thank you, Vanya. Those are excellent questions. With the first question, there's no, right now, for the majority of clonal material coming out of cryo, it has to go through tissue culture. So the exception might be dormant buds, but right now it has to go through tissue culture. So you will still have the bottleneck of tissue culture, and yes, you will still have a lag period before you can get it out. And it might be even a 3-month longer lag because it'll take 3 months to get it from cryo to get a shoot out in tissue culture. So I think that's absolutely true. And maybe that's a challenge for the hubs, is that we've got to do better at developing systems to get material from in vitro into a greenhouse, because that's really the way to go. So that's your first question. That limitation will still be there, and it's a technical limitation. I don't think it's a biological limitation right now, although biology plays a huge role. How do we know things will not degrade in the cryobank? We don't. We do have 25 to 50 years of experience, and we know that things are stable as far as we can see currently at that time frame. What I advocate is that we need to establish monitoring programs. And I think the monitoring program at the USDA in Fort Collins, and the same one that's operating at the International Potato Center with you guys in Lima, Vanya, is that you've got to take select accessions, representative accessions, and put large numbers of vials into cryo so you can afford to pull out 3 to 5 vials every 10, 15, 20 years. And this is an experiment that will run longer than your lifetime. This is something that scientists don't look at doing. But we need to do that so that the last vial, 100 years, 200 years from now, is pulled out and we can assess the viability. This is the only way we'll know for sure that cryopreservation is not causing a decrease in viability. I personally think that, yes, we will see some degradation over centuries, but it will not happen over decades. It will be centuries. But we don't know because we have not done the experiment. And those experiments should be being set up right now, today, and they are. So thank you for that question.
Thank you, Vanya. Next question. What do you think is the greatest technical challenge in bringing about the vision that you just described?
The weight on my shoulders right now is funding. I think that once this initiative is funded and we have a good year's ramp-up program, technically, we will start looking at tweaking these published research results for the crops that we're working on to get them operational. I think we will always have genotype specificity in putting things into cryo, just like we do for putting things into in vitro. However, that's going to be the minority of the collections, and I think we can get the larger in. What's going to be a challenge there is changing the dogma from short-term funding to longer-term funding so researchers can afford to look at putting hundreds of accessions into cryo and not just a dozen. So I think it's very doable, but I think the challenge will be to convert the mindsets to doing things operationally. It's just like gene banking. I mean, it's going from, it's getting people that are dedicated to the long term and not dedicated to the short term. And I think we have that in the cryobiology world today. So I think it's all very, very doable. I wouldn't be pushing so hard if it wasn't.
Yeah.
So thank you for that question as well.
The next one is, what will all this cost? So if you had them, if somebody had the money and they wanted a full bill for it, what would you say the bill is? How much do you want?
25 million for the first 5 years. That is our target in fundraising. It's based on a back of the envelope estimate on what it will cost to get this initiative up and running and get it working operationally. We already have the hubs working. The hubs are already funded at a lower level to work on the crops that are in their gene bank, but they're not funded to work on the other crops that we need to get worked on. So I think it's very doable, but I think 25 million is the target that we're looking at for the first 5 years, and that will be phase one. And it will spend all 25 million. And the 25 million, again, is targeted to the developing worlds to get those collections most at risk in the developing world safely conserved.
Okay, I'm going to merge two questions here. Can you please say what the 3 hubs are, the centers? This person is asking for the people, but maybe say the centers, who they are, and do they use the same cryo protocols for the same cultivars?
The answer to the last question is, to the best of my knowledge, yes. We'll take banana as an example, which is both in Bioversity, the main Intrust banana collection, and at IITA in Nigeria. I know that they have been working together in the development of the protocols and the exchange of the protocols. The protocols were developed at Bioversity in Belgium and were transferred subsequently down to IITA in Nigeria. The same is true for cassava, which is in Cali, Colombia, at CIAT, as well as at IITA in Nigeria. And they are talking about comparing and contrasting and sharing protocols. The potato protocol, the only gene bank in the CG, the only hub, is the International Potato Center in Lima, Peru. And that protocol has been distributed widely. There is a video out that takes you through step by step of the potato protocol so that anyone can view the video and do it. So the 3 hubs, again, are the current 3 hubs. And again, it's not cast in stone, but these are CG hubs that currently have funding for doing cryo, currently are doing cryo on an operational level, and currently have the infrastructure. They are, the European hub is Bioversity in Belgium. The African hub is IITA in Nigeria, and the Latin America hub is the International Potato Center in Lima, Peru.
Wonderful.
Thank you.
Rapid fire answers, because we want to get everybody in. What do you think about collaboration, Dave? Because if we have different centers, this person says, for different germplasm, germination, and evaluation, would that help to evaluate and collect and the collection of new accessions for this type of storage?
Collaboration is absolutely necessary, and the whole initiative is built around the hubs being the center of excellence and foster collaboration within the regions that they are. The idea is they would be there for training, for safety backup, and for doing operational cryo for those groups that don't have the facilities or the funding or the expertise. But currently, I think all the hubs are giving capacity building workshops to NARS so that NARS can come and visit and learn how to do cryo. So absolutely required. Collaboration is huge. If we don't have collaboration, the initiative will fail.
Okay, and related to that, greetings to Norma Manrique. So she asks, so the idea is to generate facilities, train people, and put up and running cryo in all countries? Or the initiative considers how many countries and crops? So what is your idea as you start this? How many countries would be involved, and how many crops would be involved?
Gracias, Norma. As you know, if we just look at the Latin American hub, in our meeting yesterday morning, which you were part of, we had 12 countries. I think that's a nice start. And I think what we want to do is be open to any country and any national program that has collections that might be able to be conserved long-term using cryopreservation. We can't do everything, so it will be funding dependent, and we will target those specific crops, initially those 10 crops that I listed, but there's nothing, again, it's dynamic. It will depend on donors and whether a donor has a particular preference. Someone comes to the table with money and says, I want you to work on X crop. We will work on X crop. And it will depend on need. What are the most at-risk collections? And what we might do is get started and going down the road doing very well, and all of a sudden there's a collection that is in dire jeopardy, and we have to shift and accommodate that collection quickly before it is lost. So I hope I answered your question, that it's open, focused on the developing world, but focused also on collections in countries most in need.
Okay. What is the most important crop to cryo most urgently right now? In your thoughts, yeah.
In my thoughts, boy, I don't have an answer to that because I don't think there's a single one crop that's most urgently needed to cryo. I think we listed the 10 target crops, and to the best of our knowledge, those were the ones that fit the program the best. If I were to say what's the one crop, I would say what are the collections, diversity collections, that are most at risk? And those would be the crops that are the priority to work on.
Okay, we've got 3 more questions.
Shoot.
Is Bill Gates interested in this sort of thing?
Who the heck asked that question? Whenever I talk to the Gates Foundation is more focused on genetic gain because that gives the biggest bang for the buck right now than conservation of genetic resources collections. And the conservation of genetic resources collections is being done by other groups like the Global Crop Diversity Trust. Would I love to have Bill Gates involved? Absolutely. I think the Bill Gates Foundation probably will be part of the funding strategy when it comes to it, but right now, conservation of genetic resources is not in their portfolio, to my knowledge.
Right. 2 more. Is there any study explaining ice inhibition for cryopreservation or the strategies and challenges? So any literature that Ahmed, released to Ahmed about this?
Yeah, I think there are lots of studies. And I think that some brilliant studies that have been done in the animal world, as well as brilliant studies on ice crystallization and ice crystal growth in plant cells. So we can explore that further. I don't have my fingers on that right now, but I would say there are people in the cryo world, plant cryo world, that could put their fingers on those papers immediately for you. So yes, that work has been done. And we know, I mean, when we do slow cooling, we'll take the vial out that we're cooling down and we'll shake it so we get quick ice crystal growth. So it's not slow. And those are tricks that are done, and lots of people know that. I just don't have that at my fingertips. Apologies.
Last question is from Matilda. Does banana include plantain? She asks because plantain is very important in Ghana. So she wonders about the hub and the cryopreservation of banana as well as plantain.
Absolutely. It does include plantain. I'm sorry, I kind of use banana generically, but yes, it does include plantain. How readily plantain goes into cryo, I don't know immediately, but if you're interested, you could ask either IITA or Bioversity, who actively do cryo with plantain. And I'm sure that that they have some success with it, if not equal success to the edible bananas.
Wonderful. So with that, Dave, I'd like to thank you very much for this wonderful presentation. We learned a lot, and the same to our participants. Thank you so much for participating today. We hope to see you in the GROW webinar in a couple of months. And as always, keep in touch. If you have any other questions, we would love to send you any more literature if you want, or any contact information. Dave, last words to you.
I just thank everybody for your interest and for participation. And thank you very much for all the questions. Yanni, Faye, back to you.
Okay. Thank you, everybody. We hope to see you again. Have a good rest of your day. Bye-bye.